Okay, next we are going to talk about the resolution, the spatial resolution of optical traps. And this is an important consideration because, of course, ultimately what we are interested is in being able to actually follow processes in biology, molecular processes that we can follow in real time. And if we want to see those molecular processes, typically those molecular processes involve a conformational change or the movement of a motor on a track or some kind of displacement of a given magnitude that we want to be able to detect with the optical tweezers. And so how well can we resolve a given displacement due to a conformational change, as I said, or a movement of a motor on a track is an important consideration. So imagine that we actually are studying a motor. For example, it could be kinesin, and is moving on a track. In this case, you can imagine this is a microtubule. And what we've done is we have attached our motor, which is shown here, like this little red guy here. We have attached it by some kind of tether, shown here in black, to a bead that is in an optical trap. So essentially, this tether becomes the transducer between the movement of the motor and the bead in the trap. And remember, since we have a good optical tweezers machine, we know exactly what is happening to the bead in the trap, how much is moving, what force is being exerted on the bead and therefore on the guy here trying to do the moving. And so, but we have this tether in between that we need to actually take into account. So the question, the rhetorical question we are posing here is, how small step can we detect? First of all, let's see what happens when we actually have a geometry such as this. And here you see an experiment, a typical experiment where actually we are studying one such motor, in this case is RNA polymerase, which is attached to a surface. This surface can be a glass surface or it can be another bead that is held on a pipette, for example. And then what you have is the piece of DNA that the RNA polymerase is going to transcribe, right? And the end of the DNA now is attached to another bead which is held in an optical trap, as shown here, okay? So what is going to happen is that when you add NTPs, the RNA polymerase is going to try to walk over the DNA, but it cannot walk on the DNA because it's attached by its back to a surface. So what it's going to do is it's going to pull the DNA through itself, and in so doing, it's going to pull the bead out of the center of the trap by an amount delta x, right? Okay. So what we want to calculate is what is that delta x that we see in this case. See, the situation is shown here mechanically in this fashion. Notice, here is the motor, the little guy here, who is about to make a step, delta x, right? So we call that the motor step or delta x step. But the guy is not grabbing the bead directly, but he's grabbing the bead through this tether, which in this case happens to be double-stranded DNA, right? Between the motor and the bead, right? So this tether or spring of DNA has its own elasticity, and we characterize that elasticity with a quantity K tether, which is the stiffness of the tether, right? Then you have the bead, and then you have that the bead itself is attached to the center of the trap, which is indicated here by the black line here, by another spring, which is the light spring. Of course, this is the stiffness of the optical trap. So essentially what we have is two springs, one due to the tether and the other due to the light, okay? And it can be shown that in this case, in fact, what is going to happen is this. You can see it intuitively. The little guy here, the motor, is going to try to make a step, delta x. But that's not going to produce a step, delta x, of the bead in the trap, because once it applies force on the DNA, part of the movement of the motor is going to be absorbed by the spring of the tether, right? And so the result of that is going to be that the bead is going to move a lot less than the actual motor. So essentially, in a sense, in this type of experiment, you lose some resolution due to the fact that your tether spring is not infinitely stiff, right? Okay, so it can be shown that the delta x that is going to be experienced by the bead in the trap, namely the amount by which we are going to move the bead from the center of the trap due to the movement of the motor by the amount delta x step, is called delta x signal and is given by this expression here. Notice it says that the signal is proportional to the size of the step, but multiplied by this quantity here, which is this ratio of stiffness. Okay? And because the stiffness of the tether is not very, very high, but rather the tether is maybe flexible, you see that in the end, this quantity, that is the ratio of the case of these stiffnesses, is going to be smaller than 1, and therefore the signal that you measure is going to be smaller than the signal, the actual step of the motor. Okay? But let's take the case where, no, in one extreme case, the tether is so soft that k tether is practically 0. Then you have in the denominator just k trap, and in the numerator you have 0. 0 multiplying by delta x step gives you a very small signal, because basically every movement that the motor makes is absorbed basically by the tether, right? Of course, DNA is not that soft, right? The double-stranded DNA is stiffer. But let's take the other example, and the other example is when we make imagine that if instead of having a piece of DNA, we will have an absolutely rigid stick here, right? Then, of course, when the little motor moves by an amount delta x, it's going to displace the bead, since that inextensible rigid tether is not going to extend, then the bead will move its full amount delta x. And you see what happens here if we take k tether to be much, much larger than k trap. If k tether is much, much larger than k trap, we can neglect k trap in the denominator, and we have k tether divided by k tether, cancels, and what we get is that the signal that we measure is identical to the size of the trap. So the result, the conclusion of this analysis is that ultimately the spring that eventually connects our optical, our trapped bead to the motor actually determines in a rather dramatic fashion the size of the signal that we can measure in an experiment like this. Let me give you an example of experiments done in that fashion. This is a system that we study in our laboratory. It's a bacteriophage Phi29, which is a virus that infects Bacillus subtilis, and these capsids of the bacteriophage self-assemble inside the host cell, and at the end the DNA has to be actually put inside the capsids, okay? And this is done by a tiny little motor that sits at the base of the capsid, and that grabs the DNA of the bacteriophage that has been replicated by the bacterium and pulls it in and packages it inside the optical, inside the bacteriophage prohead. Okay? And so you notice that we have experiment in which we have a bead in the optical trap indicated here by the tick marks, right? We have the piece of DNA that hasn't been packaged yet, right? Attached by a biotin exert-avidin linker to that bead in the trap, and we have the actual bacteriophage prohead or capsid attached to another bead by antibodies against the protein in the capsid and held there by suction through a micropipette that is introduced in the optical tweezers chamber. And now what you can do is this: you add ATP to the motor, and the motor will start pulling the DNA, and as it pulls the DNA, it's going to pull the bead out of the trap, right? It's going to start pulling the bead out of the trap, and what is going to happen is that the length of the DNA that is outside, not yet packaged, is going to be what we call the tether. The length of the tether, or DNA tether length, is going to be reducing or getting shortened in time. And that's what you see here in this second plot, where you see that in fact for 4 different phages, which are separated in time just so that you can see each one of them clearly, and also in different colors, you see that the tether length decreases in time as the bacteriophage motor packages the DNA inside the capsid. We are very interested in this motor because it's a very nice example of a ring ATPase, And as you know, cells are full of ring ATPases that carry all kinds of mechanical and transport functions. And so we are studying this motor as a system to try to understand how is that the motor actually, in this case, translocates the DNA. And this is one of the things that we observe. We observe that if we apply a small amount of force, of course, the stiffness of the DNA depends on the amount of force applied. In this case, we are using two optical traps. Notice, one optical trap has attached the bacteriophage capsids, and the other optical trap, just as I showed you before, via biotin streptavidin, it attaches its bead to the end of the DNA that hasn't been yet packaged. And using this system, we've been able to resolve the steps of the motor, and you can see that for different concentrations of ATP, and each one of these curves involves higher and higher concentrations of ATP, you see that the motor moves in this dwell manner, followed by a burst, and then dwell, followed by a burst, and so on and so forth. So it turns out that actually these dwells are the times when the motor is actually binding ATP, and the burst, of course, is the time where the motor is actually packaging the DNA, because we are plotting here in the y-axis the length of the DNA as a function of time, just as before. It's just that now we have enough resolution that we can actually see the individual steps of the motor. Before, I showed you the same plot here, but at low resolution, and you see that when you plot the length versus time, all you see is a continuum curve. But inside these curves, of course, this information about the step size of the motor. And that's what you can see here now at much higher resolution, where you see that the motor pauses for a while during a dwell phase and then translocates the DNA by 10 base pairs, and then pauses again, and then translocates the DNA by 10 base pairs, and then pauses again, and so on and so forth. Okay? Now, it turns out that 10 base pairs is not the minimal size step of the motor. This 10 base pair is made up of sub-steps of the individual ATPases in the ring. The ring has 5 ATPases. The motor has 5 ATPases, as shown here. And so the question, what we are seeing, presumably, is the result of all of these ATPases making a 10 base pairs in one cycle, one round cycle, right, for the whole ATPase. But now you may want to ask, what is the displacement caused by one ATPase at a time? And we can do that by applying a lot of tension in our DNA, right? We apply a lot of force, all the way to 40, 50 piconewtons, and now the DNA becomes very taut, very stiff. And remember, if you make the K tether very stiff, right, then you can neglect K trap here, and then it cancels, and basically the step that you measure is pretty much the step of the motor. So you get improved your resolution, and so by increasing the force, we can actually improve the resolution of this method. And by doing that, we were able to determine, using these optical tweezers, double beam optical tweezers, that the 10 base pairs burst that I show you here in these curves, in any one of these curves, are made up of 4 2.5 base pairs steps. Okay? So you see here 1 2.5, another 2.5, another 2.5, and another 2.5. And that constitutes a full 10 base pair displacement of or burst that we were seeing in this curve. So here is what we learned, for example, of this method. By using that, we know that there are 5 ATPases, but for some reason that now we are beginning to understand, we know that only 4 of the ATPases that bind to the during the dwell phase are used to translocate the DNA, so that there is 4 sub-steps of translocation of 2.5 base pairs each during the burst phase, right? Now, what happens to the 5th subunit? Well, now we know that the 5th subunit also binds ATP, also hydrolyzes ATP, but it does it for regulation. So it regulates the cycle of the other 4 ATPases. And that's data that actually is now just being published in the lab. Okay, there is another experiment that we can do for studying motors, for example, which is so-called constant force feedback experiment, right? Constant force feedback. What we do is this. We have, as before, our little motor here who is actually going to make a step delta x, right? Step. We also have the tether spring here, which may be the piece of DNA that we saw in the case of the polymerase, or could be the DNA spring that we saw in the case of the bacteriophage. We have our bead in the trap, and we have that bead attached to the center of the trap by another spring, which is the light spring. In the constant force feedback mode, what happens is this. We want to make sure that the bead in the trap maintains its position in the trap and is not displaced by the motor. And so what we tell the instrument is, when the motor tries to move the bead by pulling, right, from here, by pulling, it tries to move the bead from the center of the trap, what we want to do is we want to move the trap center by the same amount in order to prevent the bead from being displaced from the center of the trap or from the location in the trap. Because the bead is maintaining its location in the trap, the force it experiences is constant. It's always F equal K kappa, the stiffness of the trap, times delta X, whatever that displacement, constant displacement we want to maintain. And so that force is therefore constant, and that's why we call this method the constant force feedback. But essentially what it means is this: as the motor tries to pull the bead out of the trap moving, we move the trap by the same amount so that actually the bead inside the trap never moves, okay? But what it means is this, that in order to accomplish that, what you have to be able to do is to move the trap by an amount delta X step, which is identical to the amount delta X step moved by the trap. You see? So essentially, the movement of your trap, or the movement of your stage that controls the trap, is nothing but the movement of the motor, delta X. So in this case, it's equivalent to say that the effective trap stiffness is essentially zero, because you essentially move the bead by the same amount, the trap by the same amount that the motor moves. So I'm going to give you an example of this constant force displacement here on experiments that we did. This is a movie that I'm going to show you where we have a single ribosome. Let me first give you a little bit of the background. As you see here, we have a single ribosome that is about to translate this RNA hairpin, right? And so what happens is we have an RNA hairpin, and we have grouped the RNA hairpin by both ends. One end is attached to a bead in an optical trap, and the other end of the hairpin is attached to a bead atop a micropipette. And now we place the ribosome at the start site of translation, add all the components required for translation, initiation factors, elongation factors, tRNAs, amino acyl tRNAs, everything, and EFG, EFTU, and everything. And then we are going to see essentially the ribosome walking and start translating the RNA hairpin. So as the RNA hairpin actually is being translated, it's going to start open. In other words, the ribosome is going to start opening the RNA hairpin. And what we do is we have the optical trap at constant tension, at constant force. So every time the ribosome, in this case, opens the hairpin, and the system is going to become larger, and therefore the force is going to drop. The tension between this bead and that bead, the tension applied to the hairpin basically, is going to drop. The pipette actually is moved away by a given amount in order to maintain the tension constant. And what we want to see is essentially how the ribosome opens the hairpin, or in other words, how the ribosome moves during translation. And what you will see in the movie is that these dashed lines in this graph are separated by 3 nucleotides at a time. So essentially each one of them corresponds to a codon in the RNA. And please, in the next movie you will see that the motor, there it is, right? You see that the ribosome is in fact stepping in steps of 3 nucleotides at a time, codon by codon, right? Sometimes we see 2 of them together, and when the hairpin gets very close to the end, notice that the hairpin is very unstable, and then the hairpin opens completely, and we are not able to resolve the individual codons in this experiment. But early on, you can see very clearly that the ribosome, in fact, is moving codon by codon. And of course, being able to actually resolve the individual steps of the ribosome is very important because all kinds of important chemical processes are occurring here during these dwell times, and all kinds of important processes are occurring also during the translocation itself. And we want to characterize all the cycle of this motor. Well, I showed you this geometry, which we call the hairpin geometry, right? Where as the ribosome invades the RNA in order to translate it, makes the RNA longer, but the bits are pulled away in order to keep the tension or the force constant in the optical trap, and therefore we can actually follow the individual steps of the ribosome. Well, you may want to be able to play tug-of-war with a single ribosome, because notice that in this experiment, we never grab the ribosome directly. In other words, we don't know how much force the ribosome can generate. And it would be very interesting to know, to investigate the ribosome as a molecular motor, namely, how much of the energy of transpeptidation it is being used in order to actually, for example, move against a force or move against a barrier, like it could be secondary structure of RNA. So that is the geometry that I showed you here, where you can see that now we grab the ribosome itself via a biotin at the position in one of the small subunits, and which is attached, proteins in the small subunit, and is attached to streptavidin, which is attached to a bead in the pipette. And then the ribosome is attached to an mRNA that is going to translate, and this mRNA is itself attached to a piece of DNA, single-stranded DNA, to form a hybrid here that we can then connect via anti-ditch, digoxygening, anti-digoxygening to a bead that is in the optical trap. So this experiment is a tour de force experiment, because now we are going to try to observe how through this very elastic spring, which is single-stranded RNA, whether we can actually see the individual motion of the ribosome through the messenger RNA as translation occurs. Okay, and so in the next slide, you are going to see a movie of that situation. In fact, here it is. And now the dashed lines are separated by 3 nucleotides, as before, and you will see that it will start here, and you will see that, in fact, it is possible to follow very precisely by double-clicking here. You see that the ribosome starts there and now makes the transition to the first 3 nucleotides, then another 3 nucleotides, then another codon, here it is, then yet another codon. So you can see that, in fact, it is possible to follow in real time a single ribosome that is translating the messenger RNA against a force, because remember, we are now applying some tension here across the mRNA, and so we are actually loading, if you wish, the motor with some external force, and the ribosome, which is attached by the back, has to actually pull the RNA in order to actually, through itself, in order to actually be able to carry out translation. And so, doing this type of experiment, we have recently characterized the mechanochemical conversion in the ribosome motor. Okay, so finally, I want to actually discuss the issue of the resolution in optical tweezers in a slightly more formal way. And the resolution has to do with the fact that when you have a bead in an optical trap, the optical trap is here represented by this harmonic potential that you see here, this parabola, right? You see that the harmonic potential is my optical trap and holds my bead in the center of the trap. And this bead has 2 springs attached to it, as we discussed before. The light spring, which is the spring that tends to maintain the center of the bead in the trap, right? So this is related to the stiffness of the trap. And the tether spring, with which I actually grab that bead and connect it maybe to a molecular motor, as indicated here, right? The problem that I have is the following, and is that this bead that is in the center of the optical trap is subjected, as we saw, to a lot of fluctuations and noise, because we are doing these experiments in water and at room temperature. And so there is a lot of fluctuations going on. And so what I want to estimate is what is the fluctuations that I may expect at room temperature due to the movement of the bead. Well, for a system such as this, it can be shown that the bead behaves as if it would be under the effect combined of the two stiffnesses, the stiffness due to the light spring and the stiffness due to the tether spring. So the K effective that actually is determining how much noise do I actually experience in this bead in the center of the trap due to the thermal noise, the thermal fluctuations, is ultimately controlled by the sum of the stiffness of the light and the stiffness of the tether, okay? An effective stiffness. Well, I show you the spectrum of fluctuations, the mean quadratic fluctuations as a function of frequency. If you multiply it by the square of the stiffness, you can obtain the mean quadratic fluctuation in the force. And again, since that's a constant, you obtain again a Lorentzian curve, notice, flat at the beginning, then the corner frequency, and then a decay that we saw before. Okay, so here is the mean quadratic fluctuation in force experienced by the bead at zero frequency, when the frequency here, when the frequency is zero, right? Is given by 4 times kBt gamma, gamma being the friction coefficient of the bead. But notice, this curve here is completely flat, right? It's supposed to be flat here because it's a Lorentzian, and that means that you can ask, what is the mean quadratic total fluctuation that I should experience all the way to a particular frequency, say 100 Hertz. That's what we call our bandwidth of measurement, okay? So we can measure that by simply, it's just the area under this curve, is this spectrum in piconewtons square per unit Hertz multiplied by 100 Hertz. That should give me the total amount of noise in terms of quadratic noise in force. So that is expressed here mathematically as the mean quadratic noise in force that I expect due to the thermal motion for a bandwidth that is smaller than the corner frequency. Remember, the corner frequency is the place where the curve starts decaying, right? It's just nothing but what we had above, 4 kBt gamma times B, B being this frequency, which is my bandwidth, right? All right, so this is the bandwidth of my measurement, okay, in Hertz. So here is the noise. The noise that I'm going to measure is nothing of a bead in the trap. That I'm going to observe of the bead in the trap is nothing but the root mean square displacement of the bead due to thermal noise, which is equal to the root mean displacement in force due to thermal noise divided by the combined kappas, the combined stiffness of the tether and the trap, as I indicated here, right? Because I told you that this is an effective kappa or stiffness that is equal to the sum of these two. I know this quantity in the numerator because I just show it to you here is 4 kBt gamma b. So I can put it there in the numerator, take the square root because it's root mean square, and the 4 becomes a 2, and then the rest is just inside this square root, right? 1/2. Divided by k tether plus k trap. Where B, remember, is the bandwidth of the measurement in Hertz. And now I want to know what is the signal to noise ratio of my experiment. Okay, what I want is a signal to noise ratio greater than 1, which means that the delta signal that I measure at the level of my optical tweezer experiment is greater than the noise due to thermal fluctuations, right? In other words, delta signal greater than delta x root mean square thermal, right? Okay. So I know this quantity because we determined it before for the case of the little motor moving and pulling the string. Remember, you don't get the full delta x, the step, but you get delta x signal is equal to delta x step multiplied by k tether divided by k tether plus k trap. Remember? I told you that this quantity usually is smaller than 1, so what you measure as a signal is multiplied is the size of the motor, the step of the motor multiplied by a quantity smaller than 1, so you end up actually losing some resolution in that experiment. Nonetheless, we want that delta X signal to still be larger than the noise in the experiment due to thermal fluctuations. But the noise is given by this quantity, so I want this quantity to be larger than that quantity. And then that's the same thing as saying that I want the step size of the motor greater than this quantity. In other words, the motor has to be at least this size in order for me to resolve it above the noise with a signal-to-noise ratio greater than 1, right? And notice what we obtained here. We obtained a very simple relationship that we are going to see it again here. Here it is. The size of the step that we can resolve with the optical tweezers machine is given by 2 times the square root of kT, that's Boltzmann constant, the temperature of the experiment, gamma, which is the friction coefficient of the bead, all right, in water, in the viscous medium water, times the bandwidth, square root of that, divided by k tether. The first thing that you notice is that the resolution of the optical trap does not depend on the stiffness of the trap, but it depends only on the stiffness of the tether. That's the first thing to realize, okay? So there is no point in actually trying to make stiffer traps just to get better resolution, because the resolution doesn't depend on the stiffness of the trap, only on the stiffness of the tether. The other thing that you notice is that the bandwidth appears here. The bandwidth is how long are you willing to average your measurement in order to actually get a signal, right? But of course, if you make the bandwidth 0, then of course this quantity will be 0. But if you make your bandwidth 0, means that actually you are averaging your signal for infinite amount of time, and then you lose time resolution. Presumably your motor is stepping at some frequency, and you want to be able to see this process, and so you cannot average to infinite time. You have to average to time shorter than the stepping time of the motor. So that limits what can you do with the bandwidth, because if you make the bandwidth 0, of course, this quantity will eventually be 0, and this will be fulfilled for any value of delta x, but you will have no time resolution in your experiment. Okay. And the other thing to realize is that a quantity that is important is the friction coefficient of the bead. The friction coefficient of the bead, if you make the friction coefficient of the bead smaller, then this inequality will be fulfilled, and you can see smaller and smaller steps. So the idea is that you can make the friction coefficient smaller by using smaller beads. And so in general, high resolution experiments in optical tweezers utilize small beads in order to actually minimize this gamma, or the drag or friction coefficient of the bead. We are pretty close to the end of this lecture, and I just want to remind you that in fact reducing the bandwidth is great, because it means increasing the amount of time that you average your noise. So of course, if you have actually some kind of signal, as indicated here by this staircase feature here, but that is superimposed with the noise of your trap, as you actually reduce the bandwidth, you will actually average out quite a bit of that noise, reduce your bandwidth, and eventually you may be able to actually begin to see, in fact, the individual signal that you want. But of course, if you overdo it, maybe here still you can see the individual steps that are underneath corresponding to that dynamics of the motor, for example, if you imagine that that's what you are observing. But if you actually reduce further bandwidth, notice that eventually you start averaging out the signal itself, and so you lose your resolution again, because you are now losing time resolution rather than spatial resolution. So let me give you a final example so that you see the kind of quantities that we can expect to measure with optical tweezers. Suppose that you have a bead that has a 2-micron diameter, is immersed in water. The bead has a tether attached to a tether of DNA, double-stranded DNA, that is 10 kilobase pairs, and you can either apply a tension of 2 piconewtons or a tension of 20 piconewtons to that tether. The signal that you want to measure is maybe some stepping of a motor that occurs at about 1 Hertz. So that much bandwidth is required. You need at least 1 Hertz or more in order to see the stepping of the motor. The tether stiffness of the DNA can be calculated from the so-called worm-like chain theory of polymer elasticity, of DNA elasticity, and you can assume that for that, you can assume that your DNA has a persistence length of about 50 nanometers, and then you can say, well, what is the stiffness of my tether at 2 piconewtons of force or at 20 piconewtons of force? And using the worm-like chain result, you can actually obtain that at 2 piconewtons of force, your tether is 12 piconewtons per micrometer. That's the stiffness of your DNA, of your 10 kilobase pairs of DNA. And at 20 piconewtons, notice your stiffness is 170 piconewtons per micrometer. So obviously, it's convenient to work at higher forces. You can see that your K tether is much stiffer now, right? And so you want that generally. The smallest resolvable step we decided was actually given by an inequality, but then we make it equality because we are going to assume that we want just signal to noise equal to 1. So the smallest step that you can resolve is given by this amount, right? And notice that K tether is in the denominator, so having a larger K tether, the stiffness of your tether, namely using higher forces, means that you will have a much larger quantity here, which means that you can resolve a much smaller value here of the step or the distance or displacement. So we can plug all these numbers here, we know them, and it turns out that you can actually, at 2 piconewtons of tension with a 10,000 base pairs tether, which is very long. You don't need to use a 10,000 base pair. You could actually use just a 1,000 base pair, and then it will be 10 times better than that. But let's use 10,000 as in this example. At 2 piconewtons of tension, you should be able to resolve a displacement of about 1.5 nanometers with a bandwidth of 1 Hertz occurring every second, right? But if you actually work now at 20 piconewtons, notice that you can do the calculation again, and what you obtain is that you are able to resolve essentially 1 Armstrong at that 1 Hertz of bandwidth. So, in fact, optical tweezers are really wonderful instruments in the sense that they allow you to study molecular dynamic processes at high resolution, at reasonable temporal resolution, and that are of the order of displacement and resolution that are important in biological process. Conformational changes, movements of one part of the molecule relative to another, or just changes in the position of the motor in a track. Of course, this is somewhat of an ideal result because it assumes that the instrument is only limited by Brownian noise, thermal noise, and that is an important point and consideration with which we should end, and is that in as much as possible, wonderful things can be learned as long as you have instruments that actually are only limited by Brownian noise, and that's one of the big efforts in this field, to actually have instruments that are only Brownian limited and not limited by other problems like, for example, drift or temperature gradients in the laboratory or mechanical noise due to the building, vibrations or acoustic noise, etc., etc. So let me finish here and with this example, and reminding you again that optical tweezers are powerful. It's a powerful technique, but you really need to know what you are doing in order to make sure that you don't make a big mistake.